Hello, very good afternoon. Today we'll continue the topic what we did yesterday. We started with the first topic, that is an introduction to genetics. What all we discussed, let me recap it in brief. Firstly, the meaning or the definition for genetics. As I said before, genetics is a science to study all about the occurrence of heredity and the variations. Heredity is the main cause for the occurrence of similarities between the parent and the progeny, whereas the variations are the cause for the occurrence of differences between the parents and the progeny. And in continuation with the same, today we'll start with the title, that is heredity and environment. It is in fact very much interesting part of the first topic and also part of my interest, as I have obtained it from Synod and Don textbook. So here genetics is the main key component to give information about the formation of new young ones of the progeny. Gamete contributed by the parents is the only physical link between the parents and the progeny. This is very much interesting to know. Look at it, we are the kids of our respective parents. But exactly what is the guarantee or what is the evidence? Evidence is only one thing, that the gametes produced by our respective parents as well as in other all species organisms of sexually reproducing type, where it gives the only link between the parents and the offspring. This is one statement. Second statement, I like it very much, that every organism in any species, every organism is a faithful copy of its respective parents and the ancestors. Look at it, we are the faithful copy of our parents, likewise in other species, animals and the plants. Because we carry the genetic information, whatever is there in our parents is being inherited to us. That's why we are supposed to be the faithful copy of our respective parents, as well as our grandfathers, forefathers, ancestors, etc., etc. That's why every family has got its own flow of characters from one generation to the another generation till the present generation. Third statement that I like to mention is that the beginning of life starts from a biological seed, refers to be the zygote or a fertilized egg. And here this zygote, look at it, it is very much a microscopic single cell structure carrying the entire blueprint or the information of our both the parents. But that single zygote has to undergo the development, has to undergo the growth, and so as to become the new young adult individual. If you consider man as an example, we also started our journey of life from a zygote, and adult human being is literally speaking about 50 billion times bigger than the zygote from where our journey of our starting point. So we need to be very much excited to know about this, exactly how that undifferentiated, minute, very much microscopic, single cell structure could be a cause for such big 50 billion times bigger than it as a new adult body. So that way, then how it is all possible? It is all possible because of only one thing, that zygote make use of the surrounding available resources as a nutrition for its growth and the development. And here today I like to mention one more thing as an example. Like, look at it, kitten could develop into zygote of cat. That is, once it begins to develop, it becomes a kitten. Kitten develops into a cat. Elephant cub develops into an elephant. Then tiger cub develops into a tiger. Similarly, human baby develops into adult human being. Look at it, there is no change at all. For example, human zygote cannot develop into a cub of an elephant, a tiger, something like that. It is very much strange to know, but very much important at the same time to realize. So that respective, we can keep the zygotes of the elephant and the lion and the tiger and the man and the fish and the frog and the snake. Look at it. We can keep all these zygotes in the same petri and they look in the very beginning alike, more or less changing little bit size and the shape. But once they are allowed to undergo the developmental stages, then they undergo developmental stages so as to become their respective species organism only. This is all possible because that zygote contains a genetic material in the form of DNA material, DNA present in the form of chromosomes. You know, part of each DNA molecule is having n number of genes. So ultimately, gene is the point so as to enable undifferentiated fertilized egg to develop into its respective species, new young individual. This is the meaning. Then I also like to mention one more statement, that growth and development of this kind of zygote is at the cost of nature, because nature is the one to provide. That is all required for the successful growth and development of zygote into its respective species, young one. Then one more line I want to mention, heredity. You know what do you mean by heredity? Heredity is the transmission of characters from the parental generation to the every progeny and so on. And this kind of heredity operates the living individual at every walk of life, at every stage of life, through the metabolism. Why? Because body has to get exposed to the surrounding environment, and there is continuous process of repairment and rebuilding of the body. For this reason, what is required is the metabolic activities, and all the metabolic activities that occur inside every living species, living individual should be mediated by enzymatic action. And this enzyme formation is because of the gene expression. Once again, we need to come back to the root of the entire process, root point, that is the gene, and all about heredity and the variations. That way, we are directly or indirectly being regulated by overall metabolic activities, and these metabolic activities are exactly regulated timely by the enzymatic action. That way, enzyme synthesis, because enzyme is a protein molecule, for the synthesis of protein molecule, what is required is a gene and gene only. Then heredity also does one more thing, that heredity continuously interact with the surrounding environment. That way we could be able to differentiate every living individual with respect of its phenotype and genotype. Yesterday we referred these two terminologies. Phenotype is an external appearance of every living individual, and for the formation of this phenotype or external appearance of the body, internal genetic constitution, that's what we call it as a genotype. So this is all made possible because the heredity of every individual interacts with its own surrounding environment. That way it makes individual organism to look like at any given moment, and also to look differently or become differently in future. How is that? It is all being governed or regulated or determined by the heredity only. So this way, one more thing, that the phenotype of every living individual changes according to the surrounding environmental changes, whereas its internal genetic constitution or the genotype remains stable relatively irrespective of the surrounding changes. This is what I wanted to tell you. And one more, the greatest statement made by various scientists, biological scientists, like, in this world or on this earth, no two individuals may be belonging to the same species or different, do not look alike. Each one of us, each one individual organism look differently from the all others. This is all because of its own internal genetic makeup. That's what we call as genotype or the genetic constitution. This is all possible. Okay? And finally, I want to tell you that whether heredity is greater or environment is greater, a question mark. I don't want to differentiate them. They are interdependent because heredity continuously, what it does, phenotype of an individual, phenotype it is part of heredity only. Phenotype of an individual is as a result of continuously interacting with the surrounding environment. It continuously genotype interacting with its own surrounding environment. What we look today, the way how we appear to others today is all because of our internal genetic material, genetic constitution that is regularly interacting with our surrounding environment. This way, heredity is under the influence of environment, and environment influences the heredity to decide or to determine its pattern, format and everything. This way, we need to understand that every individual should have its own hereditary characters, and these hereditary characters are expressed no doubt by our genetic material, that is in the form of DNA including RNA, and also it goes in accordance with the surrounding environment. I hope you followed what all I spoke about heredity and environment. Okay? This may be asked in examination just for 2 marks, but not at all for 5 marks or 10 marks, not an essay type, but it gives you an insight to genetics or to this science, what we are going to discuss in the coming days. Next part of the syllabus, that is Mendel and his contribution to the field or domain of genetics. Gregor Johann Mendel, who doesn't know in biological students, Gregor Johann Mendel is regarded as the father of genetics. Why? You may ask me a question. The strongest foundation, scientific foundation laid by only one person in the world who is none other than Gregor Johann Mendel. Let me tell you briefly about him. Gregor Johann Mendel, born in the year 1824 in a place called Moravia, and later on he completed his early studies and became a priest for a church connected to Augustinian monastery, where that church located in Moravia of Brunn city in Austria country. Austria, please don't get it confused with Australia. Austria is different, Australia is different. Austria is a small European nation or a country which size of it is almost equal to the size of our Bangalore city. However, leave it apart, Brunn city is very much famous for this purpose, where Gregor Johann Mendel led his life and made greatest academic track records and the achievements. In the year 1857, Gregor Johann Mendel began to work on a plant called as Pisum sativum, commonly known as garden pea plant. ಕನ್ನಡದೊಳಗೆ ಇದಕ್ಕೆ ನಾವು ಬಟಾಣಿ ಗಿಡ ಅಂತ ಹೇಳ್ತೀವಿ. Pisum sativum is a scientific name. Okay. Before that, I want to tell you something about him, that he also worked as a teacher in mathematics as well as science. Though he was not a great academician in these two subjects, but he was well versed with these two. And probably his knowledge, profound knowledge in mathematics and science, it enabled him to get the wonderful experimental results. What I am telling you, that he started his work on garden pea plant for solving the question, for solving the query, something like why no two living individuals are alike. Every species, every individual looks differently. Then one more question, why the progeny of its parents look the same? Why these similarities and dissimilarities? That is nothing but heredity and environment. To give a complete meaning for two terminologies, two phenomena. One is heredity, another one is the variations. So as to answer these two, he had to work on it, and likewise he started. Why he chose this garden pea plant? You must be knowing it well, you are botany students. That garden pea plant is just a shrub, and it is a bisexual plant, very easy to handle, easy to carry, easy to grow, and it imparts the contrasting characters. I am using the word contrasting characters. Contrasting characters. What do you mean by that? Let me give you. He observed and as well as he considered about 7 contrasting characters in garden pea plant. Like how? Let me name them. First pair of contrasting characters are like with respect to height. Here I want to write traits, and here it's characters. They are contrasting characters. Trait number 1, that is height. Height could be either a tall or a dwarf. Here, tall and dwarf are contrasting to each other. One more example I gave you, position of the flower. Position of a flower. It could be axillary or terminal. I hope you follow this. Axillary, terminal. Position of the flower. Main stem ಮತ್ತು ಟೊಂಗಿ ನಡು ಬರ್ತಿತ್ತು ಅಂದ್ರೆ ಅದಕ್ಕೆ ನಾವು axillary ಅಂತೀವಿ. At the tip or apex of the stem ಇತ್ತು ಅಂತಂದ್ರೆ ಅದಕ್ಕೆ ನಾವು terminal ಅಂತ ಕರಿತೀವಿ. Position of the flower. These two are contrasting to each other. One more example. I mean, these are all the characters that Mendel considered in garden pea plant to study, to analyze, and to prove. To deduce certain principles and the laws. Then color of the flower. Color of the flower. That could be red or white. Then color of the seed. What are these? Color of the seed. That could be either yellow or green. Here, yellow and green are contrasting to each other. Similarly, shape of the seed. It could be either round or wrinkled. These two are contrasting to each other. Similarly, 6th one could be shape of the pod. Shape of the pod. It is inflated versus smooth. One more, that is color of the pod could be either yellow or green. Yellow or green. Likewise, totally 7 contrasting characters were very much visible for any person to discrete or distinguish them, and that's why Mendel made his decision to choose the right study material as a garden pea plant. For any research student or research scientist, it is very much important to select the study material or study model, like a plant model or animal model. Plant model or animal model. Okay? So, likewise. And he worked over it for quite a long time. And finally, he consolidated his all experimental results, and he prepared the paper and presented that paper in the year 1865 on 8th of Feb. Before the Natural History Society of Brunn, in front of the entire gathered scientists community, and people did not take his experimental results so seriously. However, they were accepted, and later on the book published by this Natural History Society containing all the Mendel's work and his results as well as the principles, and later on that book was circulated in European as well as American countries in the year 1866. However, to his bad luck, his research work and his research reports did not get recognition by the scientific community until his death, even after his death. He died in the year 1884, and nearly about 16 years after his death, probably in the year 1900, three scientists had to come up to rediscover the Mendel's work. So they are considered to be the Mendelian rediscoverers of Mendelian genetics. Whatever Mendel had shown and published, the experimental records, results, and the principles and the laws, they became the fundamental or basics for today's genetics, what we are learning. And those three scientists belonging to the different countries, to mention, first one, Hugo Duris from Netherlands, Karl Koren from Germany, and Schiemann from Italy, worked independent of each other in different continental countries, but came to the same conclusion that what Mendel had done before during the 18th century. However, let us get into the world of Mendel's work. Mendel worked on garden pea plant, as I said, but his method of research, his method of working on garden pea plant, in three terms I want to explain. Number one is considering pure line parents. What do you mean by this? Pure line parents. Pure line parents means now Kannadavakku Jawari anta karitheve. I am talking in terms of the plants and their seeds. For example, ಒಂದು ಟ್ರೇಟ್ ನಾವು ಕನ್ಸಿಡರ್ ಮಾಡೋಣ, ಹೈಟ್ ಅಂತ ತಗೊಳ್ಳೋಣ. ನೀವು ಕಾಮನ್ ಆಗಿ ಯೂಸ್ ಮಾಡ್ತಾ ಬಂದಿದ್ದೀವಿ. Height of the pea plant, that is stem of the pea plant, height. ನಿಮಗೆ ಗೊತ್ತು ಎರಡು ತರ ಕಾಂಟ್ರಾಸ್ಟಿಂಗ್ ಇದೆ, ಒಂದು ಟಾಲ್ ಅಂತ, ಇನ್ನೊಂದು ಡಾರ್ಫ್ ಅಂತ. ಐ ರೈಟ್ ಇಟ್ ಹಿಯರ್, ಟಾಲ್, ಡಾರ್ಫ್. Garden attached to his church, he used it for his research work. He firstly collected the seeds and grew them in garden soil as a tall plant as well as dwarf plant separately. Let me say this is a transect with only tall plants, and this is another, let me name transect A, and this is the transect B. Here he grew only the dwarf plants. What did it? You know that it is a bisexual plant, that is bee plant. Bisexual means it is having both male and the female reproductive holes in the same plant inflorescence. Okay, and the female part, you know, that is oristigma, and the male part, that is the anthers containing the male gametes as pollens. Look at it. What did it? While he grew these tall plants in one area and the dwarf plants in another area, he allowed them to undergo self-pollination. Tall plants, these are also bisexual, these are also bisexual. He allowed tall plants to undergo self-pollination for many generations, and finally he could be able to obtain all the newly produced seeds developed into tall and tall plants only, no single dwarf plant. That way, that generation he considered it as pure line tall parent. And similarly, in another garden area, he grew up only the dwarf plants, and he allowed them to undergo self-pollination for many number of generations, and finally he could be able to get the entire population of pea plants that were all dwarf and dwarf only. Like how pure dwarf plants and pure tall pea plants, he considered them as the pure line parents, so that he could be able to do the next level of his research That the second level is the hybridization. Hybridization. So hybridization experiments, the nature of his experiments are considered to be hybridization. Let me tell you, once again for our convenience, I am showing like this. This is garden area A and this is garden area B. As I said that he took pure line tall plants in garden area A, tall plants. And in the garden area B, he took dwarf plants. Firstly, what he did, he allowed the perpetuation, pollination as not self, it is a cross pollination, means anthers of tall plants must be transferred to the stigma of dwarf plants, and the anthers of dwarf plants must be transferred to the stigma of tall plants. Like how he crossed, this is called as a cross experiment. Okay, cross breeding, this also called as cross breeding. Okay, either male gamete of now ಏನಂತ ಕರೀತೀವಿ, ಪೋಲನ್ಸ್ ಅಂತ ಕರೀತೀವಿ. ಇದರ ಏನು ಫಿಮೇಲ್ ಪಾರ್ಟ್ಗೆ ನಾವೇನಂತೀವಿ, ಓವ್ಯೂಲ್ ಅಂತ ಕರಿತೀವಿ. ಓವ್ಯೂಲ್ ನಾವು ತಗೊಳ್ಳೋದೇ ಇಲ್ಲ. ಆದರೆ ಇಲ್ಲಿ ಕೂಡ ಡಾಫ್ ಬಿ ಪ್ಲಾಂಟ್ಸ್ ನಲ್ಲಿ ಕೂಡ ಪೋಲನ್ಸ್ ಇರ್ತವೆ. ಅದರ ಜೊತೆಗೆ ಓವ್ಯೂಲ್ ಇರ್ತದೆ. ಇದು ಫಿಮೇಲ್, ದಿಸ್ ಈಸ್ ಮೇಲ್ ಅಂಡ್ ದಿಸ್ ಈಸ್ ಫಿಮೇಲ್, ರೈಟ್? ಅಂಡ್ ದಿಸ್ ಈಸ್ ಮೇಲ್ ಅಂಡ್ ದಿಸ್ ಈಸ್ ಫಿಮೇಲ್, ರೈಟ್? ಸೊ ಓಕೆ, ಪೋಲನ್ಸ್ ಆಫ್ ಟಾಲ್ ಪ್ಲಾಂಟ್ಸ್ ಅಂತ ತಗೊಂಡು ಇದರ ಓವ್ಯೂಲ್ ಇದು, ಇದರ ಸ್ಟಿಗ್ಮಾ ಮೇಲೆ ಅದನ್ನ ಟ್ರಾನ್ಸ್ಫರ್ ಮಾಡೋದು. ದಟ್ ಇಸ್ ವಾಟ್ ಇಸ್ dustering of pollens from the tall plants to the what stigma of dwarf plant. In this case, ಅಲ್ಲೇನಿರ್ತದೆ ಅಂತಂದ್ರೆ ಈ ಡಾಫ್ ಪ್ಲಾಂಟ್ ಆಂಥರ್ ರಿಮೂವ್ ಮಾಡ್ತಾರೆ. ಅದಕ್ಕೆ ನಾವು ಇಮ್ಯಾಸ್ಕ್ಯುಲೇಷನ್ ಅಂತ ಕರಿತೀವಿ. ವಾಟ್ ಯು ಮೀನ್ ಬೈ ಇಮ್ಯಾಸ್ಕ್ಯುಲೇಷನ್? ಇಮ್ಯಾಸ್ಕ್ಯುಲೇಷನ್ ಈಸ್ ರಿಮೂವಲ್ ಆಫ್ ದ ಆಂಥರ್. ನಿಮ್ಗೆ ಅದರ ಡಯಾಗ್ರಾಮ್ ತೆಗೆದು ತೋರಿಸ್ಬಿಡ್ತೀನಿ. ಇಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಫಿಲಾಮೆಂಟ್ ಅಂಡ್ ಹಿಯರ್ ಯು ಗೆಟ್ ಆಂಥರ್. ಆಂಥರ್ ಶೇಪಡ್ ಇರ್ತದೆ. ಇದರೊಳಗಡೆ ಸಿಕ್ಕಾಪಟ್ಟೆ ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ಅಂತ ಇರ್ತಾವೆ. ದೀಸ್ ಆರ್ ಮೇಲ್ ಗ್ಯಾಮೆಟ್ಸ್ ಆಫ್ ದ ಪೈಸನ್ಸ್ ಟೈಪ್. ಓಕೆ, ಈ ಡಾಫ್ ಪ್ಲಾಂಟ್ ಫಸ್ಟ್ ಕೆಲಸ ಏನ್ ಮಾಡಿದ ಅಂತಂದ್ರೆ ಟಾಲ್ ಪ್ಲಾಂಟ್ಸ್ ಪೋಲನ್ಸ್ನೆಲ್ಲ ತಗೊಂಡು ಇಮ್ಯಾಸ್ಕ್ಯುಲೇಟೆಡ್ ಡಾಫ್ ಪಿ ಪ್ಲಾಂಟ್ಸ್ ಸ್ಟಿಗ್ಮಾ ಮೇಲೆ ಸುರುವಿದ. ಇಮ್ಯಾಸ್ಕ್ಯುಲೇಟೆಡ್ ಅಂತಂದ್ರೆ ಏನರ್ಥ? ಈ ಡಾಫ್ ಪ್ಲಾಂಟ್ ಇನ್ನು ಟ್ರಾನ್ಸ್ಫರ್ ಮಾಡೋಕ್ಕಿಂತ ಮುಂಚೆ ಡಾಫ್ ಪ್ಲಾಂಟ್ ಇರ್ತಕ್ಕಂತಹ ಈ ಆಂಥರ್ ಅನ್ನ ಕಟ್ ಮಾಡಿ ಹೋಗಿದ್ದ. ದಟ್ಸ್ ವಾಟ್ ಇಸ್ ಇಮ್ಯಾಸ್ಕ್ಯುಲೇಟಿಂಗ್ ದ ಆಂಥರ್ ಆಫ್ ಡಾಫ್ ಪ್ಲಾಂಟ್. ಅಂದ್ರೆ ಡಾಫ್ ಪ್ಲಾಂಟ್ ಬರೀ ಫೀಮೇಲ್ ರೀಪ್ರೊಡಕ್ಟಿವ್ ಪಾರ್ಟ್ ಉಳಿಸಿದ್ದ. ಆ ಫೀಮೇಲ್ ರಿಪ್ರೊಡಕ್ಟಿವ್ ಪಾರ್ಟ್ ಏನದು ನಿಮಗೆ ಇದನ್ನು ತಂದು ತೋರಿಸ್ಬಿಡ್ತೇನೆ. ದಿಸ್ ಈಸ್ ಸ್ಟೈಲ್, ದಿಸ್ ಈಸ್ ಓವರಿ ಅಂಡ್ ದಿಸ್ ಈಸ್ ಸ್ಟಿಗ್ಮಾ. ದಿಸ್ ಈಸ್ ಸ್ಟಿಗ್ಮಾ. ಇದಕ್ಕೆ ನಾವು ಓವರಿ ಅಂತೀವಿ. ಇದ್ರೊಳಗಡೆಗೆ ಫಿಮೇಲ್ ಗ್ಯಾಮೆಟ್ಸ್ ಇರ್ತವೆ. ಇವಕ್ಕೆ ನಾವು ಓವ್ಯೂಲ್ಸ್ ಅಂತ ಕರಿತೇವೆ. ಇದಕ್ಕೆ ಸ್ಟೈಲ್ ಅಂತ ಕರಿತೇವೆ. ಹೀಗವ ಡಾಫ್ ಪ್ಲಾಂಟ್ ಇದನ್ನಷ್ಟ ಉಳಿಸಿದ, ಇದನ್ನು ತೆಗೆದ ಬಿಟ್ಟ. ಇದಕ್ಕೆ ಇಮ್ಯಾಸ್ಕ್ಯುಲೇಟೆಡ್ ಡಾಫ್ ಪ್ಲಾಂಟ್ಸ್ ಅಂತ ಕರೆದ. ಇದ್ರ ಪೋಲನ್ಸ್ ತಗೊಂಡು ಇವ್ಗೆ ಹಾಕಿದ. ಮುಂದೆ ಅದನ್ನ ಪ್ಲಾಸ್ಟಿಕ್ ಕವರ್ಲೆ ಸುತ್ತಿದ. So as to avoid the contamination of pollen from any other plant. ಅದು ಒಂದು. ಸಿಮಿಲರ್ಲಿ ಇದರೊಳಗನೆ ಮತ್ತೆ ಎರಡು ಭಾಗ ಮಾಡಿದ. ಈ ಎ ಗಾರ್ಡನ್ ಏರಿಯಾ ಮತ್ತು ಬಿ ಗಾರ್ಡನ್ ಏರಿಯಾದಲ್ಲಿ ರಿವರ್ಸ್ ಮಾಡಿದ. ಅದನ್ನ ಹಾಗೆ ಇಲ್ಲೊಂದಿಷ್ಟು ಟಾಲ್ ಪ್ಲಾಂಟ್ಸ್ ಇದ್ದು, ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಇಲ್ಲೊಂದು 10,000 ಟೋಟಲ್ 5,000 ಹಿಂಗೆ, ಇನ್ನು 5,000 ಈ ಹೇಳ್ತಾರೆ ಈ ರೀತಿ ಮಾಡಿದ ಟಾಲ್ ಪ್ಲಾಂಟ್ಸ್. ಇವಕ್ಕೆ ಏನ್ ಮಾಡಿದ ಈ ಸರಿ ಈ ಟಾಲ್ ಪ್ಲಾಂಟ್ಸ್ ಆಂಥರ್ ತೆಗೆದುಬಿಟ್ಟ. ಓಕೆ, ಇಮ್ಯಾಸ್ಕ್ಯುಲೇಟೆಡ್ ದ ಆಂಥರ್ಸ್ ಆಫ್ ಟಾಲ್ ಪ್ಲಾಂಟ್ ಅಂಡ್ ರಿಟೇನ್ ಓನ್ಲಿ ದ ಫಿಮೇಲ್ ಪಾರ್ಟ್ ಆಸ್ ಅಲ್ಲೇ ಇದ್ರೆ ಹಾಕಲೇ. ದಟ್ ಇಸ್ ಫಿಮೇಲ್ ಪಾರ್ಟ್ ಆಸ್ ಓವರಿ ಸ್ಟೈಲ್ ಸ್ಟಿಗ್ಮಾ ಇರುವಷ್ಟ ಉಳಿಸಿದ. ಹಂಗೆ ಡಾಫ್ ಪ್ಲಾಂಟ್ಸ್ ಪೋಲನ್ಸ್ ತಗೊಂಡ ಈ ಸಾರಿ ಅವನ್ನ ಇದಕ್ಕೆ ಟ್ರಾನ್ಸ್ಫರ್ ಮಾಡಿದ. ದಿಸ್ ಈಸ್ ಹೌ ಹಿ ಕ್ರಾಸ್ಡ್ ದ ಆಂಥರ್ಸ್ ಆಫ್ tall plants to dwarf plants and dwarf plants to tall plants. Finally, he obtained the generation, and that next generation he called it as F1 generation, first filial generation. ಇವೇನು ಬೆಳೆದು ಕ್ರಾಸ್ ಪಾಲಿನೇಷನ್ ಮಾಡಿದ, ಇದಕ್ಕೂ ಕೂಡ ಪ್ಲಾಸ್ಟಿಕ್ ಕವರ್ ಹಾಕಿದ, ಇಲ್ಲಿಯೂ ಫರ್ಟಿಲೈಸೇಷನ್ ಆಗೋಂಗೆ ನೋಡ್ಕೊಂಡ, ಇಲ್ಲಿಯೂ ಫರ್ಟಿಲೈಸೇಷನ್ ಆಗೋಂಗೆ ನೋಡ್ಕೊಂಡ. ಇದರಿಂದ ಅದರ ಫ್ರೂಟ್ಸ್ ಬಿಟ್ಟು ಸೀಡ್ಸ್ ಬಂದು ಸೀಡ್ಸನ್ನು ವಾಪಸ್ ಬೆಳೆದ್ರೆ ಅದೇನೋ ಒಂದು ಹೊಸದು ಕ್ರಾಪ್ ಬಂದಲ್ಲ, he called it as F1 generation or first filial generation. ಯಾಕಂದ್ರೆ he started his experiment from pure line parents. ಅರ್ಥ ಆಯ್ತಲ್ಲ ಇದು? So this is a way how exactly he carried out experiment as hybridization experiment ಅಂತ ಹೇಳಿ. ಇನ್ನೊಂದು ಮೂರನೇ ಸ್ಟೆಪ್ ಅದೇನು ಅಂತಂದ್ರೆ allowing F1 hybrids hybrid ಯಾಕಂತ ಅನ್ನೋದನ್ನ ನಾನು ಹೇಳ್ತೀನಿ. F1 hybrids to undergo self pollination. ಈ ಸರಿ ಕ್ರಾಸ್ ಮಾಡಿಲ್ಲ, ಆಗಲೇ ಹೇಳಿದ್ನಲ್ಲ, ಇಮ್ಯಾಸ್ಕ್ಯುಲೇಷನ್ ಮಾಡಿ ಅವ್ರು ಕ್ರಾಸ್ ಮಾಡಿದ್ದು, ಅದರಿಂದ ಬಂದಂತ ಜನರೇಷನ್ ಏನಿತ್ತು ಆ ಜನರೇಷನ್ ಅನ್ನ ಮತ್ತೆ ಬೀಜ ಹಾಕಿ ಬೆಳೆಸಕತ್ತ. ಅದ್ರೊಳಗೆ ಎಷ್ಟೋ ಪವರ್ ಪ್ಲಾಂಟ್ಸ್ ಬಂದು ಎಲ್ಲೋ ಆ ಟೈಮಲ್ಲಿ ಏನು ರಿಸಲ್ಟ್ ಬಂತು ಅದು F1 ಜನರೇಷನ್ ರಿಸಲ್ಟ್ ಅಂತೀವಿ. ಆ F1 ಜನರೇಷನ್ ರಿಸಲ್ಟಿಗೆ ನಾವು F1 ಹೈಬ್ರಿಡ್ಸ್ ಅಂತ ಕರಿತೀವಿ. ಯಾಕಂದ್ರೆ ಎರಡು ಶಬ್ದ ಉಪಯೋಗಿಸಿದ್ದೆ ನಾನು ನಿನ್ನೆ, ಒಂದು ಹೋಮೋಜೈಗಸ್, ಇನ್ನೊಂದು ಹೆಟ್ರೋಜೈಗಸ್ ಅಂತ. PUC ಒಳಗೆ ಎಲ್ಲ ಕಲ್ತಿದ್ದೀರಿ, ನಾನು ಭಾಳ ರಿಪೀಟ್ ಮಾಡ್ತೀನಿ ಅಂತ ಅನ್ಸ್ಬೋದು ನಿಮಗೆ. ಹೆಟ್ರೋಜೈಗಸ್, ಹೋಮೋಜೈಗಸ್, ಹೆಟ್ರೋಜೈಗಸ್ ಲಿಫ್ಟಲ್ಲಿ ನೀವು ನೆನ್ಪು ಮಾಡ್ಕೊಂಡ್ರೆ ನಾವು ಹೈಟ್ನ ಟ್ರೇಟ್ ಅಂತ ತಗೊಂಡಿದ್ದೀವಿ ಅಂತ ತಗೊಂಡಿರ್ಬೇಕಾದ್ರೆ ನಾವು ಟಾಲ್ನ ಜಿನೋಟೈಪ್ ಇರಬೇಕು. ಟಾಲ್ ಇದು ಫಿನೋಟೈಪ್, ಎತ್ತರ ಇದೆ ಅಂತ ಬರೆದ್ರೆ ಅಂದ್ರೆ ಅದು ಫಿನೋಟೈಪ್. ಅದ್ರದ್ದು ಜಿನೋಟೈಪ್ ಬರೀಬೇಕಂತಂದ್ರೆ ನಾವು ಏನು ಮಾಡ್ತೇವೆ, ದಟ್ ಈಸ್ ಕ್ಯಾಪಿಟಲ್ ಟಿ ಅಂತ ಯೂಸ್ ಮಾಡ್ತೇವೆ. ಓಕೆ, ಈಗ ಹೀಗಿತ್ತು ಅಂದ್ರೆ ಇದು ಹೋಮೋಸೈಗಸ್. ಒಂದು ವೇಳೆ ಹಿಂಗೆ ಬರ್ದಿವಂತಂದ್ರೆ ಸ್ಮಾಲ್ ಟಿ ಬರ್ದಿವಂತಂದ್ರೆ ಏನ್ರಿ ಅದು, ಕ್ಯಾಪಿಟಲ್ ಟಿ ರಿಪ್ರೆಸೆಂಟ್ಸ್ ಟಾಲ್, ಸ್ಮಾಲ್ ಟಿ ರಿಪ್ರೆಸೆಂಟ್ಸ್ ಡಾರ್ಫ್. ಅಂದ್ರೆ here are two alleles for the character trait called height, but capital T represents tallness. Small t alleles determine height of the stem as dwarf, okay, and capital T determine height of the stem as tall, so that this looks tall plant and this looks dwarf plant. But alleles behind of that type of phenotype are present in homozygous condition, because both the alleles present on paternal and maternal chromosomes are identical to each other, and in this condition we call it as homozygous. If not, ಈ ಕ್ಯಾಪಿಟಲ್ ಟಿ ಬದಲಾಗಿ ಹೀಗಿತ್ತು ಅಂತಂದ್ರೆ ಒಂದು ಅಲೀಲ್ ಕ್ಯಾಪಿಟಲ್ ಟಿ, ಇನ್ನೊಂದು ಅಲೀಲ್ ಸ್ಮಾಲ್ ಟಿ. ಕ್ಯಾಪಿಟಲ್ ಟಿ ಇತ್ತು ಅಂದ್ರೆ ಟಾಲ್ ಆಗಬೇಕು, ಆ ಅಲೀಲ್ ಎಕ್ಸ್ಪ್ರೆಸ್ ಆದರೆ, ಸ್ಮಾಲ್ ಟಿ ಎಕ್ಸ್ಪ್ರೆಸ್ ಆದರೆ ಡಾರ್ಫ್ ಆಗಬೇಕು. ಮತ್ತು ಎರಡೂ ಕೂಡಿ ಬಂತು ಅಂತಂದ್ರೆ ಫರ್ಟಿಲೈಸೇಷನ್ ಆದಮೇಲೆ, ಪಾಲಿನೇಷನ್ ಆಗಿ ಫರ್ಟಿಲೈಸೇಷನ್ ಆದಮೇಲೆ ಆ ಸೀಡ್ಗೆ ಮತ್ತೆ ನಿಮ್ಗೆ ಗೊತ್ತಲ್ಲ, ಎರಡು ಗ್ಯಾಮೆಟ್ ಕೂಡಿದು ಅಂತಂದ್ರೆ ಹೆಪ್ಲಾಯ್ಡ್ ಪ್ಲಸ್ ಆದವು ಅಂದ್ರೆ ಡಿಪ್ಲಾಯ್ಡ್ ಆಗಲಿಕ್ಕೆ ಬೇಕು. ಡಿಪ್ಲಾಯ್ಡ್ ಝೈಗೋಟ್ ಆಯಿತು ಅಂತಂದ್ರೆ ಅಲ್ಲಿ ಕ್ಯಾಪಿಟಲ್ ಟಿ ಎಕ್ಸ್ಪ್ರೆಸ್ ಆಗ್ತದ, ಸ್ಮಾಲ್ ಟಿ ಅಲ್ಲಿ ಎಕ್ಸ್ಪ್ರೆಸ್ ಆಗ್ತದ ನೋಡಬೇಕು. ಅಲ್ಲಿ ಎರಡು ಐಡೆಂಟಿಕಲ್ ಅಲ್ಲ ಅಂದ್ರೆ this condition we call it as heterozygous. If the alleles for a particular genotype are present different to each other, then condition is called as heterozygous. If the alleles for the same character are present identical to each other, then it is called as homozygous. So just I repeated it and please bear with me. Okay, so this is how hybridization. First one, pure line parents he considered. Secondly, he carried out hybridization experiments, and thirdly, so as to obtain F2 generation from F1 hybrids, he followed a method called self-pollination instead of cross-pollination and cross-fertilization. That's how he carried out the experiments. He carried out the experiments with this type of nature, and probably these levels of research made him to get very much positive and successful results that we will see it in the next session. Just I'm going to start a short while.